Okay, so as I previously mentioned, we can classify proteins based on sequence similarity, but we can also classify proteins based on shared functional regions. So now I want to talk a bit about what are these different types of shared functional regions. So the major type of functional region within a protein is something called a domain. And this is a discrete structural unit that is assumed to fold independently of the rest of the protein and to have its own function. And it can be composed of a number of amino acids, anywhere from 20 to hundreds of amino acids. And we can have similar domains found in different proteins with different functions, because the domain is able to function independently. So we might have proteins that perform different functions, but might have a shared domain. Okay. The next type set is a set of protein sequence features. So some of these are actually related to domains, found in domains. So the first example is actually a motif. So this is a short conserved region, and these are frequently the most conserved regions of a domain. And most motifs are critical for a domain to perform its function. So the motif might be involved in an active site or for an enzyme, or binding site for an enzyme. The second type of protein sequence features are repeats. So these are stretches of amino acid sequence that get repeated a number of times along the length of the sequence. And many domains are actually constituted from repeats. And repeats may contain, again, binding sites or active sites, and they do contribute to the structural properties of the protein. The third type of sequence feature is a consensus site or a post-translation modification site. So these are conserved positions in protein sequences that may be theoretically modified post-translationally. So either by phosphorylation or glycosylation. An example is an asparagine followed by an amino acid followed by serine or threonine is a consensus site for end-linked glycosylation. So again, this is related to the possible function of the protein. So using those different features, domains as well as sequence features, we can actually use that information in order to annotate or to identify the function of a novel protein for which we do not know what domain or functional elements are contained in that protein sequence. And to do that, we use something called protein signatures. So protein signatures are computational models used to classify protein properties. And the properties that we can use them to classify are protein families, domains, conserved sites, and the protein sequence features that I spoke about in the previous slides. So protein signatures are actually built from multiple sequence alignments of related proteins. So they're built from protein, so essentially we generate multiple sequence alignments for proteins belonging to the same family or sharing a particular domain. Based on these multiple sequence alignments, we then we build a model in order to identify these conserved regions or conserved features. We then train the model by having it go through a large data set and try and predict these functional sites. And we can then use these protein signatures in order to identify functional regions or functional elements within a protein sequence of interest. So this is an overview of showing you how some of these protein signature models are built. So we start off with a multiple sequence alignment for a particular set of proteins that we know are related. So they either belong to the same family or contain a particular functional domain. Based on this multiple sequence alignment, we then look at what is conserved, what are the conserved regions in this particular multiple sequence alignment, and we build a model. And the model essentially is going to allow us to predict these conserved regions in a sequence of interest. Once we build the model, we then use the model to search through the database of sequences that we have in order to train it, so it can learn how to best predict these particular functional regions. And once the model is mature, we can then use it, and this becomes a protein signature, to identify functional regions in a sequence of interest. Okay. So there are different types of protein signatures, and these different types of protein signatures identify these different patterns that I spoke about previously. So the domains, motifs, active sites, the different sequence features that I spoke about previously. So the first type of protein signature is a pattern protein signature. And this is used to identify functional sites such as binding or active sites. And these usually consist of a few conserved amino acids. So again, as I mentioned before, these conserved patterns are identified through multiple sequence alignments of related proteins. And then once we're able to identify these particular conserved sites, we then are able to model these sites and model them using something called regular expressions. So the example I'm showing you here is, the example I'm showing you here is, so for example, this is a regular expression that we might generate for a particular active site or a particular conserved amino acid site that we observed in a multiple sequence alignment. And this is how we would represent it. So this is a D, and then in brackets we have DE and X. So what this is saying is we've identified a possible active site in our multiple sequence alignment. And this is how we represent that site in order to search against another sequence. So this site is actually a pattern, and it's composed of amino acid D, followed by either D or E, followed by any other amino acid. Okay. So this is the way in which we represent that pattern, and we can then use that pattern to be able to identify this particular protein signature or protein active binding site pattern in a sequence of interest. Okay, oops. So this is just another example of how we would generate that regular expression for the pattern. So we start off with our sequence alignment. We identify a region of interest, pattern of interest. We then zoom in and have a look at that pattern, and we look at what is the occurrence of amino acid residues in that pattern. Based on that, we then generate this regular expression that will allow us to be able to identify that pattern in another sequence. So the example I'm showing you here again is of a different type. So for this particular pattern that we've identified, we can either have an A or C, followed by any amino acid, followed by a V, followed by 4 amino acids that could be anything, followed by either an E or D. Okay, so that is a pattern that we've identified in this particular example. The next type of protein signature is something called a profile. Okay, again, it is built from a multiple sequence alignment, and it's used to model protein families and domains. So it's used to identify whether proteins are similar to a family or to identify a particular domain in a protein sequence. So a profile is built from the multiple sequence alignment and is converted into a matrix or table that describes the probability of finding a particular amino acid at a certain position. And the matrix is, the numbers in the matrix is generated based on the frequency at which an amino acid occurs at that position in the multiple sequence alignment. Okay, and I'll show you an example of it now in the next slide. But just also to keep in mind that you will come across hidden Markov models, and hidden Markov models are just a way in which we can generate a much more powerful statistical profile from a multiple sequence alignment. So again, this is just an example. So we start off with our sequence alignment. We then zoom into our sequence alignment, and then we observe what is the frequency in a particular column of a particular amino acid residue. And we then generate, based on the length of the sequence and the number of sequences we have, we can then at each position generate a scoring matrix with a frequency value for that particular amino acid at a particular position in our multiple sequence alignment. And again, we can now use this scoring matrix or this profile in order to identify a particular domain in a sequence of interest. The next type of protein signature is something called a fingerprint. So this is related back to the identification of motifs. So it's used to identify not just a single motif, but several conserved motifs. So we know that in most domains, we don't usually have just one motif, but we might have multiple motifs that need to occur in a particular order in order to carry out the function of that protein. So this is what the fingerprint signature tries to do. We identify multiple short conserved motifs, and we identify these again from multiple sequence alignments. Each motif is converted into an individual profile, so like the profiles I mentioned before, to create a fingerprint signature. And fingerprints are useful for identifying small differences between closely related proteins. So again, this is an example of how we would generate those fingerprints. So we have our multiple sequence alignment. We might identify multiple motifs across a particular region. In this case, we zoom in. We look at the frequencies of amino acids in those motifs. We generate our profiles. And then those profiles, in this case, instead of looking at the profiles independently, they are looked at in conjunction with each other when we're looking at a sequence in order to identify if these motifs occur in this particular order in our sequence of interest. So again, it's looking at these multiple motifs all together in a particular order in order to identify it in our sequence of interest. Okay. So those are that is sort of the basis of how we identify, how we can use what we know about known proteins, or where we know the function of those proteins, in order to generate protein signatures. And we can then use those protein signatures to try and identify these functionally relevant regions in a novel protein for which we do not know the function.